All right, I think we're going to go ahead and get started. I am really excited today for the Hutch at Home um, episode that we're bringing you, Life in the Lab. What do scientists actually do all day? And we're really fortunate to have Louisa Helms with us as our speaker. I'm Jeannie Chowning, a Senior Director of Science Education and Training at Fred Hutch, and welcome everybody. So Louisa Helms is a graduate student at the Freedman Lab at the University of Washington. And I think one of the great things that maybe she'll touch on too here is that she is a former student of one of our SCP teachers. And she's gone back and mentored teachers over the years as part of kind of giving back. And so she really understands the importance of education, and we've really been lucky to have her as a partner in our work. So I'm looking forward to this today, and without further ado, I'll turn it over to Louisa.
Sweet. Well, then I will quickly share my screen. Can you all see my PowerPoint?
Yes.
Cool. Okay, well, thanks, Jeannie, for the invitation and the opportunity to talk to you all. As she mentioned, I am now a graduate student at the University of Washington in the Department of Pathology. But one of the things that I think has really made a huge impact on my career is this SCP program, because I was the product of one of their students. And so just a quick background on who I am. Here's a bunch of photos of me through my scientific career, some more professional, some less so. But what was really huge in my development as a scientist, as I am today, is truly from my mentorship from my high school teacher, who used all of these classroom kits in our classroom and really got me stoked on science. I then went to Carnegie Mellon to study biology, and then I am a native Northwesterner and also had done a lab tour of the Hutch when I was in high school, and I just thought it was the absolute coolest place you could possibly work. And so I teched there for 2 years after my undergrad, and then basically knew that I wanted to stay in science and I wanted a little bit more autonomy in science. And so I decided to apply and start my PhD back in 2018 and have been working on that for now 2 years. And so what you can appreciate in some of these photos is that I have done a lot of lame things like counting cells in a microscope room in the dark for 10 hours a day and going in on Saturdays, but also getting to present my work, to take professional lab photo shoots, and then have my entire lab from the Hutch at my wedding. Science has basically become one of the most, if not the most, integral parts of my life, and I don't think I could say that had I not had such a phenomenal teacher in high school. And so I wanted to basically explore not only kind of what other people's experiences were in science, but also what their lives look like in science. Because while these pictures show you all these different aspects of my job, like if I tell you what I did that day, it's going to be totally different. And so I wanted to quickly just mention to you that there are so many different ways you can be a scientist, and these, I'm going to give you 3 examples, myself and then a postdoc and then a principal investigator. But there are so many other ways you can be a scientist, and it's not limited to just these 3. But I obviously only know my story, so I'm going to quickly tell you about what my life is like as a PhD candidate. The biggest part about what I do is I do experiments, and that can look like so many different things. I happen to work in a lab that does a lot of microscopy. So here is a little video of some of the things that I've been working on just in these last few weeks, where I've got this organoid, which I'll talk to you about in a moment. And it has this massive cyst, and I'm looking at videos of the cyst actually shrinking in culture. And so this is a recording over an hour of this cyst responding to a drug. And then using a fluorescence channel, I'm able to look at a specific protein of interest and and how it is responding and shrinking as well. And what you can really appreciate is that there's this massive bundle of green cells right there that then pull that cyst back in. And so I'm truly trying to understand why that happens. And that's pretty much my job right now. But experimental work can look like so many different things. In my own hands, I'm doing a lot of microscopy work, a lot of tissue culture work. Tissue culture work is basically just working with cells in a biosafety cabinet. But there are so many different kinds of cells that can look like different things in different hands. Right now I'm working with stem cells and differentiating them into kidney organoid structures, and then using those organoids to model different types of kidney disease. And so that is like my bread and butter. I'm making organoids every week, and then I do some kind of analysis or drug screen, and then I use microscopy to analyze that data. The other important part about my life as a PhD candidate is I'm constantly trying to understand the literature out there. And that doesn't mean like looking at a microbiology textbook. That means looking at the up-to-date literature that is getting published by fellow scientists that can hopefully shed light on some kind of work that I'm doing. One of the things that I just learned today is that you can set up little notifications to get articles sent to you every single week on a topic of your interest. So if there's something that you're really curious about, you can actually get that sent into your email every day. And if you want to know how to do that, I'm happy to show you later. The other thing that I don't think is fully appreciated as a PhD candidate that we're expected to do a lot of is attending meetings and giving talks and posters and basically just trying to talk about your work as much as possible, because communicating science is one of the skills that a PhD student and candidate needs to be able to do effectively to be able to have a successful career. The last thing I figured I would mention, my life as a PhD candidate is trying to graduate. PhDs are very long. I think the average now in the US is like 5 years. In other countries, it can be 3 years, but the US is harder for some reason. And so for me to be able to graduate and have my PhD, I have to have a first author paper published. And so to do that now, I have to do so many different experiments and publish and find something interesting enough that somebody will care about it enough to put it in their journal. And so that is basically my life right now, is trying to do something interesting that is new enough that it can be published. And so what that looks like specifically in my work is looking at polycystic kidney disease modeling in human-derived kidney organoids. And so in my opinion, one of the coolest things in the last decade that has been discovered is our ability to turn stem cells into specific tissue types. Stem cells are multipotent or pluripotent stem cells, or pluripotent cells that have the potential to become whatever they want. And what researchers have basically figured out is how to use discovery or developmental biology tools and then guide these stem cells along different lineage specifications to make certain tissue types. And so in my lab, my boss was the first person to figure out how to make kidney from stem cells, which is so cool. That will get you a professor job, just so you know, those kinds of discoveries. But now in the lab, when we have this technology, we then use the kidney organoids to then model different kidney diseases. And so what I'm specifically looking at is polycystic kidney disease, which is a genetic disease of the kidney where if you have loss of function mutations in the genes PKD1 or PKD2, you will then get prolific cyst production in your kidneys, which leads to end-stage renal failure by midlife. And what's so freaking cool about science is that we can now knock out those specific genes, turn them into kidney organoids, and then those kidney organoids without that gene actually then grow cysts in culture to fully phenocopy the disease that manifests in humans. And so what you're seeing down here are non-PKD organoids. So these have the genes intact. And then this was a CRISPR knockout edited organoid that now have grown massive cysts in culture. And so we're using these as basically like tools to do drug discovery for this disease that currently has no cure. And so
Louisa, could you clarify exactly what an organoid is for us?
Yeah, I always forget that. Thank you. An organoid is basically a dumbed down version of a human organ. And so in our case, we call it an organoid because we express 3 types of kidney cells in our organoids, but not all of them. And so it's more robust than just like a cardiomyocyte, which is just one type of heart cell, but it's less robust than a fully functioning organ. And so that's why we call it an organoid, because it's like a part way in between, better than 2D cell culture, but not quite an organ. Does that make sense?
Yeah, and then that would be useful to be able to study those organ properties in a more
Yes.
manageable way.
Yeah, one of the big caveats with kidney research is actually that if you only study kidney cells outside in a 2D cell culture dish, so like a monolayer of cells, there's so many different properties of the kidney that are lost when you then take cells and put them in 2D culture that they don't actually perform the same way they would in your body. And so the organoid kind of steps a little bit closer to what it would be like in your body in a functioning kidney.
Great, thank you.
Yeah. Yeah, so they're really handy tools, and they're really handy tools for drug discovery. And so one of the things that my entire thesis project is based upon is this discovery that was made right before I joined the lab, that if you add this drug called blebistatin to the PKD organoids, they get really big. And so you can see here on the left that these are PKD organoids that are not treated with this drug. And then once they are treated with this drug, the cysts get humongous. And so with this observation, we basically created this hypothesis that if blebistatin, which is a drug that inhibits the protein myosin, exacerbates PKD cystogenesis, or the creation of cysts and expansion of cysts, then potentially a myosin activator could inhibit this. And so this is kind of a graphical hypothesis of kind of my entire project, where the 2 proteins that are transcribed by the genes PKD1 and PKD2, they must have some kind of positive regulation of myosin contraction, which is depicted here in the schematic of the protein. And so what I've been trying to do is figure out whether or not a myosin activating drug could actually prevent PKD cystogenesis in our kidney organoids. And so I basically have found one. And so what I found is, going through a ton of different literature searches, I found a handful of myosin activating drugs, and then I screened them on our kidney organoids and found one that worked. And so here you're seeing a big picture of a bunch of different organoids in culture that were treated with our control, and then increasing doses of this drug called EMD, which had previously been studied only in cardiomyopathies and never in kidney disease. And what you can appreciate is that the cyst area, which usually is much larger at this time point, was greatly decreased with the presence of this myosin activating drug, EMD. And so this is quantified down here in the total cyst area. And you can see that there's a massive decline in the total cyst volume. And so this has basically been what I've been working on, is trying to understand, why does myosin matter in PKD? This is not a known entity quite yet in the field. And how does EMD work? And how does EMD work in the context of PKD? And so I'm trying to answer some of those questions, and hopefully that will be enough to basically grant me a passage and get a publication and get my PhD. And so this is basically what my main focus is now. But there is one more thing that I think is worth mentioning, because it's so cool, is that we can also use these organoids as a proxy for testing gene therapy delivery. So one of the biggest other things that was discovered and we've been trying to optimize in the last multiple decades is how to efficiently deliver a gene to tissue of interest. And so for genetic diseases like PKD, if you just could add back the gene, that would cure the disease like that. You wouldn't need drugs, you would just add the gene, it would function, you'd be fine. But transducing, which is the fancy word for adding a gene to a cell, is way harder than it looks. And so what we're hoping to be able to do is to use the organoid kidney model as a kind of dumbed down version of the kidney and optimize the delivery of genes in this setting that can then be scaled up into humans. And so what I've been doing the lab pretty recently is using all of these different types of viral vectors. So this is a lentivirus pseudotyped with a VSV-G envelope, and then this is a adeno-associated virus, serotype 9 specifically. And then what I've been doing is transducing them with these lentiviruses or adenoviruses and seeing whether or not I can successfully infect the kidney structure. And so this is immunofluorescence imaging of a kidney organoid. And so blue is this DAPI stain, which stains the nucleus. Green for this top image are the transduced cells. So this means that those ones were infected and now they express this green protein. Podocalyxin is a marker of the podocyte, which is the specific component of the kidney specifically. And LTL is another marker of the proximal tubule in the kidney. And so what I have done successfully now and have figured out is that I can transduce these organoids at a certain time in their differentiation process and get successful transduction. It is way harder to transduce them when they're fully mature, but if they're hit at the right time, I've been able to get good entry. And you can see the co-localization of the transduced green cells with the podocytes and with those proximal tubules. The other thing that I've been trying to do is I'll also optimize with the adeno-associated viruses. So now the podocalyxin is in green, the transduced cells are red, and the LTL proximal tubules are in pink. And you can appreciate that these are way harder to get in to my organoids. There are like 5 red cells compared to maybe 100 here. So this is a work in progress, but this is kind of like, you can imagine me pipetting virus into an organoid. That is pretty much what my life is like right now. And I think it's important to know because when I was in high school, I had no idea what it meant to actually do scientific work until I went and saw it for myself. The other thing that I really wanted to hit on this talk is that ideally we would have been able to have teachers and potentially students come and see labs. And so I did a quick recording of me touring my own lab and showing you stuff. So I'm gonna turn I would recommend the volume is very low. So if you wanna turn it up to hear it, that would probably be best. So here's a little tour of my lab.
My name is Louisa. I hope
we'll fast forward through the intro.
Give you a little showcase of some of the things that you might have missed from doing work in the lab this summer, so you can kind of see what scientists do, and when we say that we're doing something now, you can visualize what that actually means. So Courtney, if you want to follow me this way. This is our lab space. The freezes are loud, but we have -80s and -20s. And these store all of our chemicals, all of our cells, things like that. And then a bunch of glassware, a bunch of it. And then we always have liquid nitrogen tanks to store all our cells long term, or else they don't survive in the -80 if they're there for too long. If you come this way, I should also preface, our lab is one of the messier labs I've worked in, so this is a pretty messy lab, but that's okay. So we've got fridges full of stuff, so samples, medias, other chemicals, antibodies, all that jazz. If you come this way, this is my desk, and it's physically attached to a bench for good reason, so that we can work and go back and forth pretty easily. Another thing that is really important about working in science is a lab notebook. So if you want to come this way, I can show you mine. Right now I'm setting up some of the mice tissue. I'm going to mount them in these things called OCT blocks, which I set up here. And so in my notebook, I have the experiment that was done. This was a 2-page one. So it was started over here. So I've got like the date, the title of my experiment, the mice available, injection schedule, and then it's continued when I harvested them. So here are the cage numbers, all of the body weight, kidney weight, heart weight measurements, things like that. And then kind of a detailed list of what the next steps are going to be done with the tissue. The other thing that I like to do in my notebooks is like here I did a transduction experiment. So I kind of made notes to myself about the type of organ
organoid that I used. I'll show you those in a bit. What volume I was using, what type of media I was using, and then I always make plate maps because we use 96 well plates, and so this is the actual plate itself, and so you can see that this is mirrored here, and so that's really helpful. The other thing that I like to do in notebooks that I definitely learned in high school was doodling helps a lot. And so, especially for serial dilutions, I always make doodles of things and kind of show where the media goes, shows the dilution stocks at the end. And so that was something that I definitely adopted in my note-taking style. Obviously, some notes are better than others. These ones where you actually detail everything are the most helpful. These ones aren't. So if you can instill good note-taking habits early, that will be really helpful. The other thing that we do a lot of in this lab, aside from just like bench work, is a lot of tissue culture work. So if you want to come this way. So this is our main tissue culture room. And so we've got our microscope for looking at cells. I'll show you some in a second. We've got more fridges for more media because we have a million things of media. This is our biosafety cabinet or hood. I never remember the name. It's a cabinet. And so all of the stuff in here will remain sterile because there's laminar airflow that comes in, and we basically spray everything with ethanol there. Nicole, don't worry about it. You can come in. No worries. And then that I do a lot of is I make organoids from stem cells. So why don't I just show you some? Because it's more fun to see them in person than just a little picture. So that makes them real. So we've got 37-degree incubators with 5% CO2. So this maintains all of our cells, and I have a ton of cells. These are like all of mine, which is a lot. So I'm always, this is where I live. You can come through. Okay. Let me make sure the Leica is working. Okay, I'm not going to make you watch the rest because the scope was broken, so you can't see them. But that was just a quick tour so you can see kind of like how our lab is set up and how the desks are, and hopefully can give you a picture of when I say I am making organoids. Now you can see where it happens and like the plates that they're in. The other thing that I wanted to mention before I show you my next two interviews is a schematic of ways that you can be a scientist. And I think this was probably one of the things that wasn't made clear to me until I became a research technician, was that there are a million different ways to be a scientist that have nothing to do with having a medical degree. I started out as a research technician. You can go then on to be a lab manager. You can stay as a research technician forever and have an extremely fulfilling career in science, contributing to work without needing a PhD. You can be a research coordinator, so working with multitudes of labs. A lot of these people tend to work with clinical labs and facilitating clinical trials. If you feel like you want to have more autonomy on some of the work that you conduct, or if you want to basically choose the direction that your science can go and get grants on it, that's when you start to need a PhD. And so that's where I felt like I wanted that. And so that's why I decided to go to graduate school to get my PhD. And at that point, I didn't really think through too hard what I wanted to do with that PhD, and what I'm now doing during my PhD is figuring that out. And so I made a quick little list of ways that you can then go on to use that PhD. And so the natural academic trajectory is to become a graduate student, to then go on and do what's called a postdoc, which is basically postgraduate school research. In which you are not quite independent, but you're starting to gain those independent skills. From there, you can also then go on to become a staff scientist, which is, again, someone with a PhD who's working in a lab, but they're not quite like the head of the lab, but they're still extremely senior and extremely knowledgeable. And then the common academic like end all be all position would be to become a principal investigator or a PI or a professor. There's so many different words for the same position, but we call it a PI here. And that's where your name, your last name is the name of the lab. You are the one who funds it. You are the one who hires people. That would be like becoming the boss of whatever lab that you want to work in. That has canonically been the route that most people with PhDs want to go. But what I think is really cool in the last, I don't know, it's been a while, there's kind of this new field that has been more appreciated in the sciences and getting into industry. Industry basically means working at some kind of biotechnology or pharmaceutical company. The positions you can get there also range from requiring a bachelor's to a PhD. There are so many different positions, but those can be so fulfilling and have such fruitful careers as a scientist working for a company that aren't as appreciated as those in academia. The other thing that's important to mention is that being a scientist doesn't mean that you stay at the bench forever. You can go on to become a patent agent, where you then review scientists' proposals to see if they're worthy of patenting and becoming their own independent thing. You can become a medical science liaison and work with medical doctors and companies to find products for their patients that will better their lives and facilitate that. You can become a science writer. All of these articles that are getting written about the COVID pandemic are written by people with PhDs out of science journals. There are so many ways that you can be a scientist aside from just working in a lab. And I think that that's something that I really wanted to be able to express through this, is that the skills that you build in science are applicable to so many different things, and those are so important for bettering the world as a whole. The other next chunk is my interviews. So I interviewed a postdoc who I absolutely love, and a new principal investigator who also has her own core facility that she runs. And so I'm gonna move this over. And so we're gonna minimize Julie. I don't know how technology works that well, so this was the best I could do. But this is Shiri. She's a postdoc in the lab next to me, and she is going to talk a little bit about her journey into science and what her life as a postdoc looks like, because it's different as you start to do different things. So again, I would turn your volume up because it's a little quiet.
And is one of my kind of supplementary mentors that I go to for a lot of stuff as I'm going through my PhD, because she's gone through hers and has made it out. And so I thought it would be nice to kind of get her perspective on her life in science as you kind of go up the ladder in your scientific career. So Shiri, if you could do a quick synopsis on who you are, your background, that kind of stuff, that'd be awesome.
Yeah, thank you, Louisa. I think this is really great and unique, and I'm really excited that, to be part of this. So my name is Shiri. I grew up in Israel for most of my life. I finished high school, and after high school I went to the army. This is a normal military service in Israel that is mandatory. And after that, I actually traveled to America and decided to get my bachelor degree here in America. So I started in New York, and I ended up in Seattle, and I finished my bachelor at the University of Washington. But my master's and my PhD were actually in Israel, so I traveled back to Israel. I'm also married and have 3 kids, so I shifted all that to Israel, and so through my bachelor, I had my oldest daughter, and then within every degree, I birthed more and more. So my master had my Leon, and then in my PhD I had my Eli, and that's just the route that I chose. That's great. And then when I finished my PhD in Israel, when I was looking for a postdoc, of course, any place on the globe is valid and great, but because my sweetheart is from Seattle, we've decided to come back, and apply back to the University of Washington, but you know, that wasn't picnic either. You have to decide whether if you want to go to a biotech, if you want to go to a postdoc, and I'm really happy I made this decision to, you know, just settle on a postdoc. So do you want me to continue why postdoc?
Do it.
Yeah.
Yeah.
So postdoc for a mom is actually a very comfortable lifestyle, and it comes with flexibility. It comes with just hours that can be worked around the kiddies' schedule, and that worked really well for me. I'm happy to see that the world is actually transition also in the biotech field, and to be less harsh but more flexible. And now with COVID, we see that we can work from home and achieve the same amount of work, if not more. So that was one of my decisions. But what's really, really made it so unique for me, and I feel so lucky about it, that I've been exposed to science here in a way that I've never been exposed before. So my research has to do with the Institute of Protein Design. We are able to make proteins that are novel, that are synthetic proteins, and we're able to ask questions with these proteins about certain pathways in the stem cell field and the differentiation field and all that. And what makes novel design proteins super unique is the fact that one day they'll replace chemical drugs, and this is what we're here for. We're trying to make therapeutics in a much more specific and precise way, and this is one way to do it, to just become more holistic and more organic, and to just driven away and shift away from chemical drugs. My daily life in the lab are quite different than a PhD's lifestyle. Because I'm a postdoc, and because I've been through the route of conducting research, now I'm more free to just design my own experiments that one day will, you know, birth a whole new question or a field that I will hopefully one day will open my own lab with the platforms that I set at my postdoc. So that means that I can take any kind of area of research and now be more independent and conduct my own research, which is great. Part of what makes postdoc a postdoc is actually now be able to mentor many undergrads and many PhD students that have never walked this path before and might need not only the mental emotional help, but also the professional help that a PI or a head of a lab is just simply unable to do because of time. So we're the one that walking through the techniques and you know, teaching all the little tricks of how to get the science work based on our experience, which is great. Am I missing anything?
I think it would be nice, just because you keep saying postdoc, can you describe exactly what a postdoc is in terms of like how science works in the academic field, like what a postdoc means in terms of your next steps too?
Yeah. That's a good point. I never really defined that.
Yeah.
So a postdoc means that now there is just a different avenue of research. So as a postdoc, I write my own grants, and this is true for PhD too. I write my own grants, but I also try to collaborate with a lot of people to get network and see if I can, you know, learn more or distribute my science more. So let me tell you about my science, and then I'll tell you what that actually rolling into. So I was super fortunate to work with novel design proteins that remodel the epigenome, which are big word, epigenome, but it just means that it's just beyond the DNA. Any protein that is beyond the DNA and acts on the DNA is at the epigenome. So we've created this synthetic protein. And what was really cool in my postdoc, that I was able to write a patent with the University of Washington and have it under my name and my PI names, and not only to roll it into a real patent, but also have it licensed at the biotech company. And one of the things that I'm really super excited about, and that's something that happened in a postdoc, is that I can take this patent and actually roll it and spin out a biotech company. And that's something that I'm really putting all my energy and efforts to just take this technology and see if we can now translate that into therapeutics and to really drive it to better medicine. So that's one example of how my postdoc looks like.
Yeah. I'm gonna pause it there because I know that I'm running out of time and I want to get to the other interview as well. So that was Shiri. She's a postdoc. She does really cool work. This is now Julie, who is a principal investigator and head of a few of the different cores at the lab that I work at. So this is someone who's basically done her postdoc and now started her own lab. So she's like one rung higher on the like lab hierarchy. The people I go to for mentorship, aside from my own mentor, and she's just a wonderful all-around person. So, Julie, do you mind telling us just a little bit about yourself and kind of your background?
Yeah, sure. So, like Louisa said, I'm a director of the Ellison Stem Cell Core at the Institute for Stem Cells and Regenerative Medicine, and I'm also an assistant professor in comparative medicine. And I'm also the co-director of the UW Mouse CRISPR.
So everything.
Yeah, a lot of things. Yeah, how did I get there and what's my background? So I'm, like you probably can hear, I'm from France. So I did my PhD in France and in cancer, and looking at programmed cell death, apoptosis, and leukemia. And then I came here in Seattle for a postdoc. Working on pluripotent stem cells and cancer, and I'm trying to understand when we put cancer cells in hypoxia, if they start expressing some some stem cell markers, and why they are so aggressive in that cases. And then I from being a postdoc, then I transitioned to acting instructor and then assistant professor here at UW.
Is that a pretty typical
transition? So very often people after the postdoc are trying to apply for a lot of different faculty positions. In my case, I wanted to stay, so I got 2 kids also in between, and I wanted to stay in Seattle. I really love Seattle. My husband had a job here, and so I tried to find ways of staying here and keep doing and working with all the people I'm working with. So I had several options, and I really hesitated to going and be more a research scientist in a lab or really be a PI. And that was a big struggle for me because, and we can talk more later about this, but there's this life-work balance also. And when you're a PI, you do a lot of other things. And so,
can I stop you too?
Of course,
I think I didn't even fully understand what PI meant until I started working in a lab. And so if you could explain the difference between being a scientist, for like a staff scientist versus a PI, so that they can understand that, that'd be awesome.
Of course, So a PI is actually a principal investigator. So you direct a lab. Research scientist, you're under a PI and you do a lot of technical work mostly. I mean, you also, as you climb the ladder as a research scientist, do a lot more than just experiments. But a PI really needs to manage a lab, write grants, and has a lot of other obligation with the university, be part of a lot of committees and a lot more managing role.
When you are a PI and you're the director of the core, and
Another core.
And the CRISPR core.
Mouse CRISPR core. We do transgenic mice with my
how do you
department.
do all of that? Like, how?
That's a good question. I don't know. We
And
manage.
you have 2 kids.
So technically on paper, I'm supposed to do 50% of my own research and 50% of the core.
Okay.
On a day-to-day basis, I cannot separate like this, 50%,
No.
50%. That's not how it works,
Yeah.
right? So when we have a lot of work in the core, and maybe I should explain a little bit what we are doing in the core
Yeah.
also, so you can understand a little more what we do. So the core is actually, and we can take a little tour after that, but we have 3 tissue culture room where we rent space and hoods for people to come and do pluripotent stem cell work. So we are a pluripotent stem cell core.
Mhm.
And so everyone in UW, in ice cream, but also outside children's or others, more like the whole Seattle area,
Mhm.
when they want to learn about pluripotent stem cells and they don't have any expertise in their lab, they come here.
Oh,
We
cool.
have the
Okay.
expertise and we can teach them and show them how to culture those cells. So we have some trainings to culture pluripotent stem cells and embryonic stem cells. We have, we know how to reprogram skin cells or blood cells into pluripotent stem cells. That's called reprogramming.
Mhm.
And so we teach that also, or we offer it as a service as well. So we have a lot of services also where we do a lot of genome editing.
Are
So-
you doing that, or do you?
So Jen and Chris,
Okay.
so I have 2 technicians, Jen and Chris, that are doing most of the
handwork.
work with the cells, but I do all the design
Okay.
of all the CRISPR experimentals, the genome editing experiments.
Okay.
So usually investigator contact me, they have those cells that they got blood cells from a patient that have a disease and they want to try to study this disease in a dish.
Mhm.
So what they ask us to do is to take these blood cells, reprogram them into pluripotent stem cells,
Mhm.
and then correct the mutation if they know where the mutation is. Very often it's just one base pair that is
Yeah.
mutated. So then we use CRISPR-Cas9, a genome editing technique, to correct that mutation in the pluripotent stem cells. And then we give them back their cells, and then they can, in their lab, differentiate them into
Whatever.
cardiac cells or a lot of different things, and then study them without having to do all this background work to have the lines that have the mutation and have the corrected mutation.
Yeah. 'Cause that
So
is
we offer this.
cumbersome.
So we offer this as a service, and we also test a lot of reagents, try to test some new methods when we have time because they are pretty. We have a lot of work right now.
Yeah.
So, which is cool. It's
Yeah.
great.
And then how does that differ from your work in the mouse core? Is it
Yeah.
still pretty similar, but now you're doing CRISPR transgenic mice?
Yeah, exactly. So since I'm part of the comparative medicine department, I work with transgenic facility in UW. And people want to make transgenic mice to knock out conditionally, for example, a gene in the mouse to study a human disease, for example. And so they come to me and they have no idea what to do. They don't know anything about their protein.
Okay.
Or their gene at least.
Okay.
And so what I do is I look at the gene, I come up with a strategy, I talk to them about it, we come up with a plan, and then we have 2 solutions. Either we go directly in the zygote, so the 1-cell embryo of the mice, and do the correction with CRISPR directly in this 1-cell embryo, and then reimplant in the mouse to try to generate a transgenic mice. When we want to do very big changes, we found that it's easier to still go in the mouse embryonic stem cells here in the core.
Okay.
We do those mutations, and then they can be injected into the blastocyst of an embryo and then generate chimeric mice.
Cool. Okay, so those are your basic core positions.
Yes, exactly.
And then your PI work, how does that fit in? Because that sounds like a lot.
Yeah, so I have several projects actually that I'm doing. So I'm focusing mostly on understanding, it's very broad, but understanding cell fate changes in normal situation or pathological situation, and in particular, I'm very interested in cancer. And so I study early development, human development. I've done recently a whole genome CRISPR screen to understand the first step of human development. But I'm also following up different genes that are involved in cancer and try to take those pluripotent stem cells, differentiate them, have mutation, and try to do 3D cultures to try to understand those cancerous gene as well.
So in terms of, I know you're an assistant professor, right?
Yep.
So in terms of how your lab works right now, how would you describe that?
Yeah, so it's actually pretty recent that I became the director of the Licensed Stem Cell Core. It's just this month, at the beginning of the month. So I'm kind of transitioning. Until now, I do a lot of collaboration with another lab here, the Rural Baker Lab. So we were kind of sharing the space, and I was doing most of my work over there. Now I transitioned to do most of it here.
Okay.
So we have we can walk there, but we have a tissue culture room, but we also have one room where we can do experiments with a bench and everything. So I'm doing all my work there now.
Are you physically doing it, or do you have employees yet?
Right now, I'm doing it because my two technicians are working for the core, and it's a cost center, so I cannot really have them work on my. But I just got a grant from Fred Hutch, and so I got a little bit of money to hire a technician. Okay. And I used to have undergrad students. Right now it's the summer, but they're gonna come back. And so I'm gonna build my small little team, and I will probably do less myself.
Good. And then, in terms of how PIs kind of grow, in terms of where you're at, where do you see your lab going in the future? Do you anticipate keeping the director roles of the cores and just kind of having a little lab on the side?
Yeah, that's a good question, and we'll see. Like I said, it's very recent, so we'll see how it evolves. As it is now, it's a very small room, so it's okay for a few people to work there. As I grow, maybe I will still do a lot of collaboration with Alma, so maybe we'll share some area, but we'll see. Maybe I will have to expand and have another space as I get more grants. It's all about grants also. So one other thing that the PI is spending a lot of time doing is writing grants to get money for research and for hiring technicians, so have students. So we first need to get money. Yes.
Yeah.
And so once we get more grants, then we can ask for more space.
Yes. So you were saying earlier that 50% of your work is technically supposed to be core, 50% PI. What do you think the actual breakdown is in terms of your day of work?
Yeah, it's really depends. Right now, I would say it's been like 70% core. Yeah. Because also with all the COVID situation, for a few months we didn't do as much work in the core. So right now we have a lot of projects piled up, and so we're trying to catch up.
Okay.
But otherwise, I'm hoping it's gonna be close to 50-50 at some point.
And then you said that you did your PhD in France and then you came here. Yeah. Is there a reason that you wanted to come to the US?
Yeah, so France, it's interesting how we do research in France.
I'm going to pause there. I'm going to fast forward, everything she's saying is great, but we're running out of time. And I want she did a quick tour of the lab, and I want you to see what it looks like, because it's totally different than what mine looked like. So this is the tour.
I'll follow you.
Sorry if you get dizzy. Yeah, and if there's anything cool that you want to show us in the scope, that'd be awesome.
Okay, yeah, we have some a cool micro-injection scope. It's actually the one that we are using for embryos.
Oh, let's do it.
And for mouse embryos, when we in transgenic, we have the same machine. It's like, I'll show you, using joystick.
Oh, yeah.
So here is actually the part that is my lab, and where we do all the molecular biology work as well when we do genome editing.
Yeah.
And so here the bench, and we have a fume hood and some UV lamp to do a look at the PCR, the DNA, and everything. And right here, so that's a core. Here we have one of the tissue culture room with Jen over here.
Again.
With one of the technicians in the core with passaging some cells right now.
Get it.
The way it's set up is we have all those tissue culture hoods here where we do all our cell work, and incubators to store our cells here. So cells are stored at 37 degrees at 5% CO2. And we have in every room microscope, centrifuge, and everything that we need to culture those cells. And actually, the pretty cool piece of equipment I was mentioning earlier,
Oh my gosh.
is right here. So you see, you have your little joysticks here.
Oh, do those control the arms?
Yes,
Oh my
exactly.
gosh.
So you have needles here, and so you can have embryos and inject DNA or cells in those embryos.
Oh my gosh.
So that's pretty cool.
Yeah. That's awesome.
And that's actually exactly the same one that we're using in the transgenic facility when we do all those transgenic work in the lab.
Do you also, okay. That's it. Sorry. I hope you could hear everything. The main take home that I wanted you to get from this was that all of us who are basically all called scientists do so many different things, and they can look like so many different things, and you can be from Israel or France or wherever, and you can still do such cool work. So I hope that came across. I'm gonna stop sharing my screen, and if there are any questions,
please put them either into the chat or feel free to unmute yourself.
Yeah,
And
I see the one in the chat from Tim. Cell counting isn't lame. True. Very good point. Yeah. Does anyone?
yeah, what questions do you have, students especially? Or there's a few teachers I see here as well. Love to hear questions you might have. Sometimes it takes a minute for the questions to come through.
Yeah, no worries. The other thing I should mention is that I only talk to people in academic science. And so I hope that also came across, that this was entirely skewed towards working in the University of Washington in my stem cell area. And so working at the Hutch is totally different. Working at different campuses is totally different. And so it's just amazing the breadth of work that you can do in science, even from lab to lab.
Mm-hmm. So you may have heard the acronym ICE cream that was mentioned in one of the videos. So it's the Institute for Stem Cell and Regenerative Medicine. And it's actually in South Lake Union near Fred Hutch. And there's a lot of collaborators, like you heard the PI talking about her work with Fred Hutch and getting grants from Fred Hutch. So there's a lot of work that happens in both institutions, a lot of overlap.
The other thing that I didn't show, just because I see there's no questions, so I'm going to keep talking, is I asked both Shiri and Julie kind of what their passion for science kind of stemmed from. And Shiri's was from high school. It was from doing dissections in a high school lab class. And Julie's was a TV show in France where it's some cartoon body system where everyone is animated and the immune system goes and fights the evil things in the body. It's those little things that if you are sparked by them, it can be a whole career for you. And so it's so important that if you feel lame in your science class and you think it's lame that you like it, it can be so cool later that you should stick with it.
One of the things that came up, I think a lot in your talk that I haven't seen so much in other talks, is kind of the international aspects of science. So could you talk to that a little bit about, seeing folks from other parts of the world and getting to interact with them and all of that?
Yeah, science is it's like math. It's a language that is universal. And one of the things that's so cool about working in science is that if you can fund yourself, you can work anywhere. And so like Julie and Shiri were mentioning, to get grants, that essentially is your way to do science. You have to be able to write and communicate science effectively to then be able to do it. And so if you are from anywhere, you can do science. And one of the coolest things about doing science in the United States is that it is extremely well funded compared to other countries. Shiri was mentioning that in Israel, it was much harder to get grants. In France, Julie was having issues basically being able to become a PI because the positions are so much more limited. And so in the United States, we get a lot of really talented people coming here to work, and you get exposed. I remember in my last lab, by only being able to speak one language, I was the most limited in my ability to communicate because everyone else knew at least one or two other ones. It's so cool. Hanako, is that how you say it? Sweet. How do you think working as a research tech before starting grad school shaped your grad experience so far? Oh, it was vital. One thing that I really didn't know when I finished undergrad was what the heck I wanted to do next. And starting graduate school right after sounded so scary because it was such a long degree to commit to, that I wanted to work for a little bit before and figure out if doing research all day, every day was something, one, that I could do, and two, that I would enjoy. So doing that, I took two years to work at the Hutch after undergrad, and it was just so pivotal in my ability to figure out what I wanted to do and whether I wanted to stay in research science or go somewhere else. I could not recommend doing that enough.
I'm curious, like the students who are on the call, was this kind of new information for you about the different kind of ways to be involved with science at different levels? I know people use the terms a lot, but it's like Louisa was saying, people don't really stop to break it down very often.
Yeah.
Oh, so there's a question about how did you know you wanted to work with stem cells?
Oh, I took a class in my undergrad career, and it was just all about stem cell engineering. And I thought it was the Nobel Prize came out, I think it was in 2008, for Shinya Yamanaka, who basically figured out how to turn a skin cell into a stem cell. And that basically just exploded this new field that I knew of. And then I took that class and I actually got to learn about research that was being done with that information. And I listened to his Nobel Prize acceptance speech, and it was so amazing. Just him talking about the potential that this had for the future of medicine, that it made me so giddy to think about being able to work on something that could be like the next wave of how science works. And so that's how I went and I worked at the Hutch in a virology lab after that. And then I worked in 2 years studying HIV, and still that passion and that spark for stem cells was still there, that I then found the Institute for Stem Cell and Regenerative Medicine at UW, and just wanted to work there because I thought it was just the freaking coolest thing ever. Tim, might have missed it, but are you working in the same lab for your grad program as you worked in your gap years? No. So I think I just briefly mentioned, I worked in the Emmerman lab at the Hutch, and they are like a 30-plus-year-old lab with a really senior PI who has been around since we didn't even know how HIV worked. And he was one of the pioneers in understanding how HIV works in the human system and how our bodies combat it. It was so cool to work in that lab. I got to work with live HIV virus and like make HIV in this negative air pressure room. It was so cool, but it wasn't something that I felt like I could actually make a difference in human lives doing. It was a very basic science lab, and there are different types of lab work that you can do. And the thing that I really cared about was doing translational work that could potentially have an impact on human lives, and doing that preclinical work that I'm doing now. All right, Jeannie.
Okay, yeah, so unfortunately we're out of time, but if folks are interested in having more questions for Louisa, please send them to me and I'll send them on to her. And I just want to remind you that we have a schedule up at our Scp News and Events for upcoming talks every Tuesday, 4 to 5. So hopefully we're on repeat in your calendar. And next week, Miranda Lehman from the Chapuis lab at Fred Hutch, she's a graduate student who's going to be talking about immunotherapy. So playing hide and seek with cancer is the name of her talk, and talking about harnessing people's own immune system to fight cancer, and how cancer evades sometimes that mechanism from our immune system. I also want you to, if you can, fill out the feedback form, give us some feedback. Put the link into the chat, and there's a QR code there. If you would take a moment, give us some feedback, that'd be great. And we hope we see you next week. Can you please unmute yourself and give some chat reactions and some clap icons for Louisa. Yay, thank you.
Thanks, Louisa.
Thank you.
Thanks, Louisa.
Thank you, Louisa. Great job. That was awesome. Thank you.
Thank you, guys.
Yeah, so great. Thank you, Louisa.
Bye.
All right.